Welcome to our webinar, where we will be taking a deeper look into the incorporation of the recently released OECD QSAR Assessment Framework into ECHA's evaluation of QSAR studies in REACH registrations. I'm Adam Elwan, your host today. So here's the program. We'll be starting with Tomasz Sobański from ECHA, who will be giving you an overview of our activities to support the phasing out of animal studies. Then, Patience Brown, the Principal Administrator of the OECD's Hazard and Exposure Assessment Programs, introduces you to the OECD QSAR Assessment Framework. She will be followed by Doris Hirman and Andrea Gissi from ECHA, who will be sharing a presentation on our perspective on the QSAR Assessment Framework. They'll talk about our current practice in assessing QSARs under dossier evaluation and how they compare with the framework. They will also take you through upcoming changes in EUCLID to address the new framework. I will then conclude with a few take-home messages, followed by a 15-minute break before we start the live Q&A. You can send your questions throughout the event by going to slido.com and using the event code QAF 2024 or with the QR code on this slide. Your questions will be answered during the live Q&A. If you want to address a question directly to one of the panelists, add their name to your question. Now, we can only cover topics within the scope of this webinar. So if you have questions that are not directly related to the topic, you can send them to us using our contact form at echa.europa.eu/contact. A Q&A with answers to all the questions will be published after the event. So if your question is not answered by the time we close, you'll find it in the published Q&A. The presentations are already available on our website, and the video recording will be there right after the event ends. As mentioned in the previous slide, we will reply to all the questions we receive. So those we have time for during the live Q&A and the rest in the published Q&A document. If you want to stay up to date with our latest news and events, subscribe to our newsletter at echa.europa.eu/subscribe.
Welcome. My name is Tomasz Sobański and I'm leading Alternatives Method team at the European Chemicals Agency. And today I will give you the brief introductions to ECHA efforts towards phasing out animal studies and why QSAR Assessment Framework is an important part of it. But before I will move to more details about it, let me start with the beginning. Why we are promoting alternatives to animal testing? First, because we are mandated by the legal text of REACH to do so. Article 1 of REACH is talking about promotion of alternatives method. Then, just to give you the short recap of what REACH is about, REACH registrants, so people which are importing or producing chemicals which they want to put on the European market, needs to provide information about ecotoxicological properties. Those informations are part of so-called standard information requirements. But REACH as well gives the registrants the opportunity to adopt some of those information requirements using alternatives methods such as in vitro tests, read-acrosses, or QSAR's predictions. And criteria for those adaptations have been listed in Annex 11 of REACH. ECHA, from our side, what we are doing, we are working on development and promotion of alternative methods by providing the guidance, and QSAR Assessment Framework is one of those examples, by developing new ways of characterizing hazard, and by contributing to the discussion about future regulatory system when basically there will be no need for animal testing, or at least reliance on those animal testing will be limited significantly. But if you will look at what is the current status and how alternatives have been used so far, we can see that adaptations already now are used more than experimental studies, and that that read-across is the most used adaptation. However, what about QSARs? QSARs at the moment are used in less than 3% of all properties submitted within the REACH registrations. If we look more closely on what type of information requirements have been covered by QSARs, we can see that the QSARs were used mainly in bioaccumulation, in aquatic toxicity properties, all type of aquatic toxicity properties, and in skin sensitization. However, we have an impression that registrants could consider more QSARs in their registration dossiers, and in addition, some of the QSARs which have been already submitted are not of the good quality. Therefore, there is clearly room for more effective use of QSARs under REACH. And that was one motivation why we wanted to have QSAR assessment framework. But talking about minimizing the use of animal testing, first we can say in the short-medium term, the extrapolation from existing toxicological knowledge is probably the most effective way. Reliable QSARs are probably more suitable for covering simpler endpoints, while well-justified read acrosses can be used for more complex ones. But this will not eliminate absolutely the use of animal testing, because some part of the chemistry is simply not tested yet. So we cannot extrapolate from existing knowledge if we don't have it. So for those parts, to further eliminate the need for animal testing, we need to invent new ways of characterizing hazard without the need of animal testing. And for that, let me briefly introduce you ECHA idea on ECHA approach towards animal-free hazard assessment in 3 steps. First is to define what are the critical needs, what are the critical elements which we need to cover scientifically and practically to enable this transition to non-animal testing. This is very important because it will allow us to focus research developments on those areas. In the same time, in parallel, in step 2, we can demonstrate that actually some of those existing techniques can be used already now under current legal framework. Trying to use them to support some categories, to cover certain endpoints in certain conditions will build our confidence in how how NAMs and different alternative methods can be used in practice. Finally, knowing what are the critical needs and being able to cover them, plus gaining the experience in how we can use them, we can think on step 3, which is a redesign of the current system to make it more adjusted and more aligned with and compatible with new approach methodologies and new way of characterizing hazards, which will not require animal testing. But today I will focus only on the step 2 because QSAR assessment framework is one of the elements in this step 2. So talking about demonstration, we identify 4 main areas. In silico I will leave for the end. But on the top of use of in silico methods and QSARs, we as well foreseen use of molecular data for read across and grouping with clear acceptance criteria, meaning to give, like we did with QSAR assessment framework, to as well give clear indications how different types of molecular data or mechanistic data could be used to justify and substitute the read across cases. Then we believe that there is a need to establish robust practices for in vitro and PBPK, in vitro PBPK and toxicokinetics modeling to allow better characterization of industrial chemicals. It is not possible at the moment with all industrial chemicals due to the fact that their properties vary a lot, and not for all of those properties we can do it properly at the moment. Finally, there is a need to integrate omics into regulatory toxicity testing to have this opportunity to use molecular data in the regulatory context already now. And then talking about in silico methods, here we foreseen 2 actions. One is to making more data available from the REACH registrations, so new data which were not used so far in building the models, and make them available to the developers in order to give them opportunity to do the new models with broader applicability domain and with higher accuracy. This is the first step to make QSARs more widely used in the regulatory context. And the second part is exactly QSAR Assessment Framework, which means giving clear indications what reliable prediction means and what are the criteria for valid model. So let me focus now on the QSAR Assessment Framework and why we believe this is so important part of our strategy and on our work towards animal-free future. So first of all, QSAR Assessment Framework comprised of two main elements. One was related to validity of the QSAR model. This is not entirely new concept. It was already, OECD had already validity criteria for the models. They were refreshed and refined, but they were existing before. What is completely new in the QSAR Assessment Framework are the reliability criteria for the prediction, because we know that even the best model can give you the wrong predictions if your chemical is out of the applicability domain or simply the chemical space is not covered well by the model. So we believe that being explicit on what reliable prediction means will first provide the guidance to QSAR developers that they can implement those criteria in their tools and they can indicate it directly to the users whether this prediction is reliable or not. This will allow users to as well assess the reliability of the predictions, even for the tools which were done before in the older tools when you will not have it done automatically, because you can do it as well yourself applying the criteria. And finally, we believe that it will bring you the transparency how we assessing the QSAR results and other authorities are assessing the QSAR results once they are submitted, and how we are using them in our regulatory work. So then basically we believe that there is a very direct and straightforward path from the new criteria into the wider use of adaptations, because clear and transparent criteria is a key for wider acceptance of QSARs and model predictions, both by users and by authorities. Wider acceptance of QSARs means that means more new regulatory applications. There will be more consideration where QSARs can be directly used in the different legal frameworks. And more regulatory application will lead to significant reduction for tests, needs for tests, and reduce the cost of your registration efforts. And with that, I would like to thank you, and I wish you very nice rest of the seminar. Thank you very much.
Good day to all. My name is Patience Brown. I am the Principal Administrator for the Hazard Assessment and Exposure Assessment Programs at OECD, and it's my pleasure to be here today to give an introduction to the QSAR Assessment Framework and also provide a little bit of context on where we see the QSAR Assessment Framework fitting in the OECD vision of new approach methods. So there are a number of global drivers to use new approach methods and alternatives to animal testing in chemical risk assessment. These include increasing the throughput of the number of chemicals that can be tested, as well as, in many cases, increased relevance to the target species, which is often humans. Of course, there's the reduction in animal use, but there's also a reduced time to making chemical safety decisions and being able to use the best available science in doing so. OECD considers new approach methods primarily in two project areas. The first of these is the OECD Test Guidelines program. OECD test guidelines are internationally harmonized methods for generating data to evaluate chemical hazard. Many of these include NAMs, primarily in vitro approaches, and though not exhaustive, you can see a list here. So not only are the test guidelines themselves, but there's also guidance documents on how to use and interpret the resulting data. OECD test guidelines are validated following principles that were described in Guidance Document 34, which is the validation and international acceptance of new or updated test methods. And the results of these test guidelines are covered by the mutual acceptance of data, meaning that regulators can accept the data generated by OECD test guidelines if they're conducted following quality assurance practices, and there's no need to retest. So there's a high level of confidence behind the resulting data. In addition to the Test Guidelines program, the OECD Hazard Assessment Program considers how to use new approach methods as well. This group is primarily discussing approaches and practices for integrating information to come to a regulatory decision on chemical hazards. So rather than how to generate the information, really how to combine the information to come to a conclusion. Some of the projects under the hazard assessment group that use new approach methods include the case studies using integrated approaches to testing and assessments, or IATAs. These are demonstrations on how one can combine multiple different new approach methods to come to a conclusion. Also, the guidance document on chemical grouping and read across. The third edition of that document should be available in 2024. There's a number of projects and a subgroup of experts that discuss how to use omics approaches to support regulatory decision-making, and then a variety of various topic-specific guidance documents that are proposed, developed, and then declassified and publicly available. The Hazard Assessment Group is really a forum to discuss how to build confidence in new approach methods and identify the aspects that can be harmonized. Also under the Hazard Assessment Program are a number of interlinked tools to support regulatory decisions made on chemicals. These are collectively referred to as the OECD ecosystem of electronic tools, and starting on the left side, they begin with the OECD Harmonized Templates, or OHTs. OHTs are standardized data reporting formats for not only traditional toxicity test data, such as in vivo tests, but also new approach methods like mechanistic and intermediate effects. OHTs can also be used to report exposure and use information. And I should say that there are OHTs for every OECD test guideline, but also the OHTs can be used for non-test guideline data as well. So structured data leads to structured databases and greatly facilitates data sharing. The structured database can then be used for a variety of interrelated tools to query and access the information. For example, eChem Portal can be used to search for property information from various chemical databases. Also, the Adverse Outcome Pathway Knowledge Base, for example, can be used to access the structured data and build predictive models. Of course, such data also contributes to regulatory data submission and use, but I'd like to particularly highlight the QSAR Toolbox, which is a user of such data and a key analytical tool. The OECD QSAR Toolbox work began in the early 2000s, and it was developed initially with a goal to support chemical category and grouping approaches. Using data from a tested category member or an analog, one can fill data gaps for the untested members of the same category and reduce the need to test the entire group. Well, certainly the QSAR Toolbox is that, but now it's also much, much more than that. Included in the toolbox are experimental data for almost 60 databases that include 100,000 chemicals and 3 million data points, which can also be used to contribute to grouping and read across approaches. There are also a variety of profilers that predict activity based on chemical structure or mechanism of action. And there are metabolism simulators and experimental databases that predict not only effects of the parent, but also the downstream metabolites. So all of that is to illustrate how the QSAR Toolbox supports alternatives to animal testing and is a key feature of the OECD NAM strategy. The QSAR Toolbox can be used to inform testing strategies by forming categories and identifying data gaps, or in some cases filling the data gaps. When additional data need to be generated, intelligent testing strategies can be designed to reduce costs and the number of animals required. The QSAR Toolbox can predict properties and in many cases replace information requirements or be used as input to support authority assessments. The QSAR Toolbox can also be used from the very earliest stages to support green chemistry. The toxicity of substances can be predicted even before they're produced. So all of that provides, hopefully, context for where the QSAR assessment fits. In order for regulators to use QSAR predictions in supporting dossier submission or prioritizing chemicals for additional test needs, one needs to have confidence in such approaches. So the objective of the QAF was to develop a systematic and harmonized framework for regulatory assessment. The scope includes not only QSAR models, but also QSAR predictions and results based on multiple predictions. In terms of the relevance and applicability, the QAF is meant to be independent of the technique used to build the model, the specific endpoint predicted, and the intended regulatory purpose. So it's meant to be quite broadly applicable. The audience was thought to be primarily regulatory authorities, but it's also a very valuable reference for other stakeholders using QSARs for regulatory risk assessment, as well as for QSAR model developers. The QSAR assessment framework, like all OECD projects, represents a consensus opinion of international experts. The project was added to the OECD Hazard Assessment Work Program in 2021, and it was led by ISS Italy and by ECHA. But it was also supported by a very committed group of international experts on QSARs. This QAF expert group provided general input on the project and feedback on the proposed path forward, as well as written comments on drafts. They met through a series of teleconferences and a face-to-face meeting as well, and contributed to what we now have as a declassified document that was available in Q3 of 2023. And the QR code is available for you in the lower right-hand side. So the QSAR assessment framework was based on some existing guidance. Guidance document 49, which describes principles for validating QSAR models, and guidance document 69, which was guidance for validating the models themselves. In other words, how to apply those principles. And I should note that both of these were built around or adapted from guidance document 34, which are the principles and approaches for validating test methods for international acceptance. So as a first step, the group evaluated this existing guidance and found that all of the principles for assessing models were still relevant, but they developed new sections on principles for assessing resulting QSAR predictions and assessing results from multiple predictions used in combination. So in addition to these new principles, the group also developed assessment elements for each principle and a checklist of criteria. This includes guidance for how to determine if the criteria are met and some examples illustrating how to evaluate the criteria. All of this contributes to a much more transparent and systematic way of validating and assessing the scientific rigor of QSAR models, predictions, and results from multiple predictions. For those who would like to learn more, I encourage you to visit the OECD QSAR Toolbox website. You can see the website here, which is easy to find via Google, but also a QR code. The QSAR Toolbox website includes recorded video presentations that introduce the QAF in more detail. I know you'll hear some of that information later in today's webinar, but this is probably a higher level introductory information. There are, of course, links to the QAF and background documents, as well as links to how to use the QAF in a regulatory context. So with that, I thank you all very much for your attention. For those who are interested in learning more about the OECD programs or about the new approach methods discussed at OECD, here's various references to get a hold of me or find out information via the web. Thank you.
Hello, my name is Doris Hirmann. I'm working in the Alternatives Methods team at ECHA. Patience just gave you a brief overview of the QSAR assessment framework in the OECD context. And now we are looking a bit more on the REACH perspective, particularly on the REACH dossier evaluation process. So we have divided the content into 4 parts. I'm going to talk about general aspects, such as what type of information you can find in the QAF, how to use the checklists, and also what is the difference between a QSAR prediction and a QSAR result. My colleague Andrea Gissi will give you more details on practical aspects in the other 3 parts. The QSAR Assessment Framework is a guidance for regulatory assessment of QSAR models, predictions and results based on multiple predictions. If you are interested in a more extensive information on the QSAR assessment framework outside the REACH context, there is a 1.5 hours OECD webinar available, which was recorded in November 2023. And you see here on the right side the QR code where you can access the YouTube video. So why did ECHA engage in this activity? QSARs are allowed in many chemicals regulations, and so also under REACH. REACH promotes the use of alternative methods to vertebrate animal testing, and QSARs can be used to fill data gaps if they fulfill certain conditions. There are the OECD QSAR principles that cover the scientific validity of the QSAR models, which was published in 2004, and also a guidance was published in 2007 on this topic. However, the use of a valid QSAR model does not yet guarantee the validity of each result. And on the right side, you see an illustration to explain this. We do have actually in ECHA guidance in place on the assessment of QSAR models and QSAR predictions. So it is the Guidance on Information Requirements and Chemical Safety Assessment Chapter R.6, QSARs and Grouping of Chemicals, which was published already a while ago in 2008. Still, there was a need to establish the principles to assess individual results and to have a systematic and harmonized assessment framework for QSAR models and predictions. And to do this at OECD level is fantastic, as we now have a framework where we can refer to, and not only us at ECHA, but around the globe. My colleague Andrea Cissi is going to talk about the comparison of our current approach and the approach by the QAF later in this webinar. So ECHA did this work to help, in the first place, us as authorities to assess QSAR predictions and QSAR results. But we think it will help also QSAR users when preparing their QSAR predictions and results to, for example, fill data gaps under REACH. During the discussions of the QAF, the QSAR assessment expert found that the OECD principles for evaluating the scientific validity of the QSAR models are still relevant. Those are the 5 principles. First, a defined endpoint. Second, unambiguous algorithm. Third, a defined domain of applicability. Fourth, appropriate measures of goodness of fit, robustness and productivity. And 5, mechanistic interpretation if possible. So they stay the same. So what is new in the QSAR assessment framework? There are 4 new OECD principles for evaluating QSAR predictions and results based on multiple predictions. There is number 1, the correct input. Then there is number 2, substance within applicability domain. Then there is number 3, reliable prediction. And number 4, outcome fit for purpose. And there was a reason why to introduce the principle number 3. Actually, applicability domain already addresses to some extent the reliability. However, there is no universally agreed applicability domain check, and each model developer can define as they see need how to check the applicability domain. There may be other checks important for the authorities, and they are captured then under reliable prediction. And I will talk more about this a bit later in the webinar. Now let's see what elements there are in the QAF guidance document. So first of all, there's the text document, which establishes principles for assessment of QSAR results and explaining how to assess models and their results. We have here as parts of the document the foreword, executive summary, visual abstracts, and I will explain a bit more in the next slide on that one. Then we do have 3 chapters or 3 sections that address the checklists. So we have 1 checklist on QSAR models, 1 on predictions, and 1 on results derived from multiple predictions. Then there is final considerations, and we also have in the Annex 1 the QMRF, which is a minor update of the previously already existing QMRF. There is, I think, only 1 field about the description added. So in most cases, the the current QMRFs, they should remain valid. Then we have in the Annex 2 a QSAR prediction reporting format, QPRF in short, and there was a major update done to represent now the 4 new principles that were established. And then you also see a glossary of selected terms. Now, this one is a screenshot of the visual abstract that you can find in the QAF document. It should illustrate the different roles. So we have the QSAR model that then result in a QSAR prediction. And we expect the model developers to establish the QMRF that should give us all the necessary information to address the QSAR model. And then we have the QSAR user, which is responsible to fill in the QPRF or to provide the information equivalent to a QPRF that should address everything that is related to the QSAR prediction. And then we have now the checklists under the new QSAR assessment framework, where we can assess with the model checklist the information given in a QMRF. And we have the QAF prediction checklist that we can use to assess the information given in a QSAR prediction reporting format. So for us, we expect that will be a really big help, but we think that also for model developers and for the users, these checklists may be really a good source of feedback if all the information is present in their material that is needed by the authorities. Now there is in the QSAR assessment framework not only a Word document, but there is also an Excel document that contains several sheets and will help in practice to assess QSAR models and QSAR predictions and the results. So let's have a bit closer look on what is in these Excel spreadsheets. So first of all, you have the checklists for the model, prediction, and the results. But the spreadsheets are more than just the checklists. You have there also separate spreadsheets that provide details, practical advice, examples, and the mapping to the QMRF and QPRF for each assessment element. There is also a section dedicated on how to assign uncertainty levels for predictions and for the results. Now, where can you find all these documents? So here on this slide you have the links. You have a glossy version and a mono version available of the Word file. You have the Annex 1, which is the QMRF, and the Annex 2, which is the QPRF. And also quite interesting, it's available as a Word file that you can fill in. And then you have the checklist as Excel spreadsheets. Now let's go a bit more into detail of the QAF. So I said in the beginning that there are 5 principles for the evaluation of QSAR model and 4 principles for a QSAR prediction. These principles are further divided into assessment elements, which we abbreviate with AE. And you can imagine these assessment elements as building blocks. So the higher certainty you have on the single building blocks, the stronger the house is that you can build with them. Ideally, all the building blocks should be strong and reliable for a stable house, but in some circumstances it may be sufficient to build quickly a shelter, and not all the building blocks may be equally important. So this means, depending on the purpose of use, evaluators may accept models where not all the assessment elements are fulfilled. Now for each of these building blocks or assessment elements, there is guidance given. Underneath there is a paragraph, and here you can see an example for the first principle for assessing a model, which is defined endpoint. So assessment element 1.1, clear scientific and regulatory purposes. So you have the same here for the QSAR predictions. You have the 4 principles broken down in different assessment elements. And again, you have here guidance given for each of the assessment elements. And the guidance also explains the conditions for acceptable predictions. It's good to read through the text, but actually the checklists, they should have all the necessary information that should be enough for you then to perform an assessment. So let's go now to these checklists. We have here a screenshot of the checklist, and there is the principles and the assessment elements listed. And for each of these assessment elements, you can fill in the weight. The weight is intended to report how important this assessment element is in the context of use of the prediction. So it depends on the purpose of the use of the prediction. There are default values given, high, medium, low, but it can be changed by the assessor. Then you have the outcome. It can be fulfilled, not applicable or not assessed, or not documented. And for each of the assessment elements, you can also assign uncertainty, low, medium, high. And then you can still give comments. The uncertainty was added to help the assessor when there is not so clear-cut cases. So maybe in some cases, you don't know if you should assign fulfilled or not fulfilled, is quite black and white. So with the uncertainty level, medium or high, you can then still add information about this and maybe make a prediction acceptable even if the assessment element is not totally fulfilled. So the checklist for the QSAR model looks similar, but the weight and uncertainty are not in this list. And then for each of the predictions, you have a field to add the conclusion on the individual prediction, the uncertainty of the prediction, which can again be low, medium, or high, and it is based on the highest uncertainty of high-weight assessment elements. And then you can add the outcome of the assessment, which is acceptable for the intended purpose, not acceptable for the intended purpose. Documentation is insufficient to decide on the acceptance for the intended purpose. And the document suggests to accept predictions with low or medium uncertainty. And then there is again the possibility to add the comments. Now let's have a bit closer look at the 4 principles. And for the first one, I would like also to present an example to illustrate how you could work in practice with the checklist. So the first principle is the correct input. It should be complete and representative of the substance being analyzed and uses reliable parameters. And this input is not very trivial when you think of rich substances which may contain different constituents or impurities. So to get the input right is not trivial at all. Then here we have 3 assessment elements. So there should be a clear and complete description of the input and the model settings, so that in principle, if we want to reproduce the prediction, that we are also able to do so. Then the input should be representative of the substance under analysis. And the third is reliable input parameters. So there are models where you can add manually parameters. For example, log KW can be optionally added. So this third assessment element addresses this. Now let's have a look how you can use the checklist now to fill in or to assess these 3 assessment elements. Here is a screenshot of the spreadsheet prediction criteria and uncertainty. And on the left side, you see the principles and the assessment elements, and then you have different colors. So the first column, it says what to check and how. The second says practical advice. Then you get examples, and there is also a column on the uncertainty. And what you cannot see on this screenshot, there is on the right side more information. It gives also examples how to assign the uncertainty. And here is the text that is in the spreadsheet. So what to check and how. For the assessment element 1.1, is it clear whether the structure is input using smiles or other identifiers? If other parameters are also used as input, they are described. And if relevant conformational information is also given, in case of editable options, check if default settings are applied, and if not, if a justification is provided. Now, to make that more understandable, there is example given. A model requires smiles and optionally log KW as input to generate a prediction. Is the assessment element fulfilled? If yes, assign the uncertainty. So it gives their instructions. Low uncertainty, the smile and the log KW are provided. Medium uncertainty, smiles is provided, log KW is not provided. High uncertainty, only the cast number is provided, but cast and smile association is ambiguous. There is also practical advice given. So if the input is incomplete, but the assessors are still able to reproduce the prediction, then the weight of this element in the overall assessment is lower. Now let's go to the second principle. This is about the substance is within applicability domain. And this is here limited to the domain as defined by the model developers. And now I prepared one slide to illustrate a bit the applicability domain, because it is also could be quite complex. So you can have applicability domains defined very simple, for example, only the molecular weight range, but it can be also much more elaborated in different layers. For example, the mechanistic domain, parametric domain, metabolic domain, and so on. So what is an applicability domain? It is the applicability domain of a QSAR model is the physicochemical, structural, or biological space, knowledge or information on which the training set of the model has been developed. Predictions outside the applicability domain have higher or unknown uncertainty compared to predictions inside the applicability domain. And predictions within the applicability domain of the model are more reliable than predictions outside the applicability domain, as illustrated here below. Under this principle 2, there are two assessment elements. There is the substance within applicability domain. That means the substance meets the applicability domain requirements specified by the model developers. But there is a second assessment element added to capture also any other limitations of the model. So sometimes there is in some general help files, there is additional limitations mentioned. To capture also this, there was an extra assessment element introduced, specifically to address the other limitations. Let's go now to the principle 3, which is the reliability of a prediction. This was introduced to capture any important element that is not yet addressed under the principle 2, which is the applicability domain as defined by the model developer. So this principle 3 will give the authorities the means to assess anything that they think is important to address a reliable prediction. So it covers elements that may not be part of the developer's definition of the applicability domain. And it has quite many assessment elements. So it has as first the reproducibility, That means that the authorities should be able to reproduce if they want the result. We have the element 3.2, overall performance of the model. If it is possible to assess how well the model performs when predicting substances that are similar to the one under analysis, then information on the overall performance of the model would become of lower weight. Then there is the third assessment element, fit within physicochemical, structural, and response spaces of the training set of the model. And then the next one is performance of the model for similar substances. I think this is quite an important one. So the overall model performance is assessed, but it gives more confidence to know that the model also predicts well chemicals which are similar to the one to be predicted. Then the assessment element 3.5 is about mechanistic and/or metabolic considerations where relevant. So maybe it's less relevant for screening, but if the information about a specific mode of action is known or formation of specific metabolites, then this needs to be taken into account when assessing the acceptability of the prediction. And the last one is about the consistency of information that can be within one endpoint or also between endpoints that are related. My colleague Andrea Gissi will give you more details in the following presentation about the QAF and our current practice under REACH. Now the fourth principle, it is outcome is fit for purpose. And this is about the usefulness of the computational prediction to answer a specific regulatory question. There are three assessment elements in the fourth principle on the outcome fit for regulatory purpose. 4.1 is about compliance with additional requirements. An example are regulations that require the use in combination of two different types of models to produce a result, such as in the ICH guideline M7, assessment and control of DNA reactive mutagenic impurities in pharmaceuticals to limit potential carcinogenic risk, which requires the use of one expert rule-based and one statistically based model. So in this case, the use of an individual prediction will not be fit for purpose. Then there is the assessment element 4.2, correspondence between predicted property and property required by regulation. Then there is the assessment element 4.3, decidability within specific framework. So this is about allows us to check that the result is expressed in a form or a unit that can be compared with regulatory thresholds. For example, if we want to assess biodegradation half-life, or the endpoint is about biodegradation half-life, and a ready biodegradability test result in form of BOD, or like a prediction in form of BOD is presented, then this does not match. And also it includes a check that there is sufficient level of confidence that the prediction lies on one side of the threshold. There is an example given on BCF. So if we have the threshold of, or the criterion BCF of 2000, if the predicted BCF is 200, it's well below the threshold, and then it allows us to decide on the criterion. But if the BCF is very close to the threshold, the uncertainty is high, then we may not be able to decide, or we cannot decide. Now let's move to the last part of the presentation, of my presentation, which is on the assessment of results based on multiple predictions. And here is actually a screenshot of the glossary, and it explains what is a QSAR prediction and what is a QSAR result. A QSAR prediction is an individual output of a QSAR model, and it can be continuous or categorical output. while the QSAR result is the assessment of a property of a substance based on multiple QSAR predictions. And here is a flowchart to illustrate how to work with these QSAR results. So the first step is to fill in for each individual prediction the result checklist. So you may have either more predictions for one substance or one chemical, or you may have predictions of, for example, different constituents and with the same model. Then the second step is to check how the final result is determined, and there is an additional assessment element. It is the correct determination of the final result from individual predictions. Then next you determine the uncertainty of the final result by weighing the uncertainty of individual predictions, e.g. consistent independent predictions have lower uncertainty. And then the last step is to decide on the acceptability of the result for the intended purpose. That means that for one purpose, the result may be acceptable, for another purpose not. The document suggests to accept results with low or medium uncertainty. That brings me to my last slide, the take-home messages. So the QSAR Assessment Framework was published in August 2023. It establishes new OECD principles for assessment of QSAR predictions and results from multiple predictions, and provides guidance and checklists for their assessment. The QSAR Assessment Framework becomes a reference point for regulatory assessment of QSARs. And with a systematic and harmonized assessment framework, the QSAR Assessment Framework will benefit regulators first, but also followed by model developers and QSAR users. So thanks a lot for listening. And the next speaker is Andrea Gissi, who is going to present more practical aspects from an ECHA perspective. Thank you.
Hi, I'm Andrea Gissi from the Alternative Methods team in ECHA. Doris has presented an overview of the QSAR Assessment Framework, while with me we will go more into the details. Let's start with Part 2, ECHA's current practice in assessing QSARs under dossier evaluation. We have presented in detail how we assess QSARs under dossier evaluation in a previous webinar that is available on our website at the link shown in the slide. I'm going to summarize it in the next slides and then compare our practice with the QAF. Rich requirements for using QSARs to adapt standard information requirements are specified in Annex 11, 1.3. There are 4 criteria. The model must be scientifically valid, the substance in applicability domain, the results adequate for purpose, and the documentation adequate and reliable. We explain in ECHA Guidance R.6 how we do the assessment. And the guidance makes reference for the scientific validity of the model to the OECD QSAR principles, for which a guidance was published in the year 2007, which are that the model must have a defined endpoint, an unambiguous algorithm, defined applicability domain, and appropriate measures of goodness of fit, robustness, and predictivity. When we assess the scientific validity of the model, as I said, we refer back to the OECD QSAR principles from 2007. To assess if the substance is within applicability domain, we check the applicability domain as defined by model developers and the parametric, structural, mechanistic, and metabolic domain as relevant. To verify that the results are adequate for purpose, we check the input structure and the reliability of the prediction. For the documentation, we assess the QSAR prediction reporting format and the QSAR model reporting format, or equivalent content. Is the model scientifically valid? For the defined endpoint, we assess the quality of the data used to build the model, which is the training set. Concerning the algorithm, we check that the prediction is reproducible, meaning that the same input and settings lead to the same output. We check that the applicability domain is defined. We check the availability of measures of performances, but we do not check the mechanistic interpretation, because this is given as a not mandatory principle in the OECD document. It says, if possible. To assess whether the substance is in applicability domain, our starting point is the definition of applicability domain as provided by the model developers. However, we also consider the following aspects: the descriptor domain, the structural domain, the mechanistic domain, and the metabolic domain. When it comes to adequacy of the results, we have to define the purpose, and here we are talking about adequacy for adapting rich information requirements. For the input structure, we check that the input structure that has been selected is correct, and this is not trivial in case of multi-constituents or substances with unknown or variable composition, complex reaction products, or biological origin, because in these cases more than one structure might be relevant for the prediction. When it comes to the reliability of the prediction, we check the reliability of the input parameters in case those were input manually by the user. We check the presence of analogs in the training and test set and accuracy of their predictions, and the consistency of predictions with other information available for the substance. When it comes to the documentation, the model documentation must be provided as a QMRF or at least contain the equivalent information to it, which means information on the predicted endpoint, including data on the experimental protocol and data quality for the data used to develop the model. We want to see an unambiguous definition of the algorithm, descriptors of the models and its applicability domain, and an estimate of the goodness of fit and predictivity of the model, including information on training set and validation statistics. For the prediction, the documentation is provided in the form of QSAR prediction reporting format, which should include the prediction and the endpoint, a precise identification of the substance modeled, information on the relationship between the modeled substance and the defined applicability domain, and then identities of close analogs, including considerations on how predicted and experimental data for analogs support the prediction. So these were our practices before the QSAR assessment framework, and as of today we still assess QSARs according to the practice I have just presented. In part 3 of my presentation, starting now, we will compare our evaluation practices with the OECD QSAR Assessment Framework. Let's start from the scientific validity of the model. As I said, when assessing models, ECHA refers to the OECD QSAR principles from 2007. And as Doris has explained in her presentation, the QAF expert group confirmed the use of principles from 2007, with special attention to quality of data used to build the model. But we already checked data quality under the first OECD principle, a defined endpoint, in line with the assessment elements 1.2 and 1.3 in the QAF model checklist, transparency and quality of the experimental data. Second, REACH requires the substance to be within applicability domain. What we do in our practice is to refer to the applicability domain as defined by the model developers, and also consider the parametric, structural, mechanistic, and metabolic domain as relevant. However, the QAF considers under applicability domain only what is defined by model developers. But if we look carefully, under reliability, the QAF checks in the assessment element 3.3 of the prediction checklist the fit within physicochemical, structural, and response spaces of a model training set. and in the assessment element 3.6, the mechanistic and/or metabolic considerations. When we look at this information together, we could consider the two approaches as aligned. Third, REACH requires the results to be adequate for the purpose. And what we do in our practice is to refer to the input structure and reliability of the prediction. On the other hand, the QAF checks that the input is correct as a first principle and reliability as a third principle. So let's look at the reliability in more detail in the next slide. Here on the left, we have what we assess under dossier evaluation, and in the column on the right, we have the assessment elements from the prediction checklist from the QAF. We can see in the first row there is the input structure that we check under reliability, and this is covered by the assessment element 1.2, input representative of the substance under analysis. Then we check the reliability of input parameters, which correspond in the QAF to assessment element 1.3, reliable input parameters. Then we check the presence of analogs in training and test sets and accuracy of their predictions, where in the QAF we have a corresponding assessment element 3.4, performance of the model for similar substances. We have in dossier evaluation the consistency of prediction with other information available for the substance, which corresponds to the assessment element 3.6, consistency of information. And then in the QAF we have more requirements, the assessment element 3.1, that is reproducibility. However, in dossier evaluation, we consider under the unambiguous algorithm for the validity of the model, the reproducibility of the result. In the QAF, we also have the assessment element 3.2, overall performance of the model, that we check under the validity of the model. And then we have the assessment element 3.3, fit within physicochemical structure and response spaces of the training set of the model, and assessment element 3.5, mechanistic and/or metabolic considerations that are considered under applicability domain in ECHA's practice. So as we can see from this table, when looking at reliability, the dossier evaluation practice and the QSAR assessment framework consider exactly the same elements. Finally, REACH requires that the documentation is adequate. And what ECHA requires is information on the model as a QSAR model reporting format and on the prediction as QSAR prediction reporting format, the QMRF and the QPRF, or equivalent information. And the QSAR assessment framework provides updated versions of this document. So also in this case, we are aligned. Okay, so in short, what changes in ECHA's assessments with the publication of the QAF? The scientific assessment will not change, as the QSAR assessment framework is fully aligned with the ECHA current practice. And this did not happen by chance. ECHA brought its experience to the OECD group, and then this experience was formalized in the QSAR assessment framework. When it comes to our decisions in compliance check, for now, we will keep using reference to the ECHA guidance R.6. However, in future, we will refer to the QSAR assessment framework to be even clearer on the identified issues. And this leads to the last part of my presentation, which is about the EUCLID changes triggered by the publication of the OECD QSAR Assessment Framework. So the changes in EUCLID are based on the updated QSAR prediction reporting format in the QAF. There are 17 new fields that appear when the user selects as a study type QSAR and assigns a reliability score. The benefit of these new fields are for the user, because they help the user to report the key information needed for a QSAR, but also facilitate assessors in finding key information for evaluation. And in this way, we will have information that is structured in EUCLID fields rather than in attachments in terms of QPRF, information on the prediction. When it comes to the model information, the QMRF, we did not create new fields, and the QMRF we still expect it as an attachment. especially if information is not easily publicly accessible. Otherwise, you don't need to submit it, we can go and find it online. So what are these updates of the QPRF and the QMRF? The QSAR prediction reporting format has been updated to version 2.0, and this is a major update to reflect the QSAR Assessment Framework guidance. It has 8 main sections: the general information, substance, model and software, prediction, input, applicability domain and limitations, reliability assessment, and purpose of use. So you can recognize here that after the first general information, then we have the assessment elements and the principles from the QAF reflected in the sections. When it comes to the QSAR model reporting format update, this was updated from version 2.0 to version 2.1. So it's just a minor update. There is only one new field and more details in the description of how to fill in the fields. And this is why this is because the OECD principles for validity of models have not changed, and therefore we felt that there was no need for a major update of the QMRF, also to facilitate the work of QSAR model developers who have already put a lot of effort in creating their QMRF, and with such a minor update there is no extra work for them. Here we can see a screenshot of the new fields in Euclid. On the left, we see the more general information about model name and version, software name and version, and remarks. And then in the results, there are fields to report the applicability domain and the justification and other elements that you will recognize from the QSAR Assessment Framework. Some of these fields are free text fields, but some other fields are pick lists, such as the fit within the applicability domain, so that you can just select one of the options available there. These fields will be available in Euclid in the major release planned for April 29 this year. We expect registrants to fill in the new Euclid fields for QSARs when they prepare new dossiers, or if they need to update dossiers for other reason, but these new fields will not be mandatory for now. But by completing the new fields, it is easier for registrants to make sure that the relevant information is reported in the dossier and also considered by us during dossier evaluation. And with this slide, I would like to thank you for your attention.
Thank you to our presenters. Before we start the live Q&A, I'd like to touch on some key points regarding the latest developments at ECHA and how they impact you. So my first take-home message today is that our current practice under dossier evaluation has now been reflected in the OECD QSAR Assessment Framework guidance and checklist. As mentioned by Andrea, our scientific assessment of QSAR studies remains consistent. Now, we're committed to maintaining high standards in this area to ensure the reliability of our assessments. Moving forward, I'd like to remind you about the important change in how we communicate in compliances. We're now referring to the QSAR Assessment Framework to provide even clearer explanations when rejecting a QSAR study. This ensures transparency and helps you to understand the reasons behind our decisions. Lastly, I want to emphasize the importance of following the guidance, new reporting formats, and the Euclid fields. These resources are there to help you in providing all the necessary documentation for our assessment. By adhering to these guidelines, we can streamline the process and ensure a smoother experience for everyone involved. These updates represent our ongoing commitment to maintaining high standards and fostering transparency in our regulatory processes. Now, it's time to start our live Q&A. Before I hand over to our panel, here's a reminder of how the Q&A will run. You can send us questions through Slido using the QR code on this slide, or by going to slido.com and using the event code QAF 2024. The live Q&A lasts until 2, after which we end the webinar. If your question is not answered, you can refer to the published Q&A that will be coming soon. You can also get in touch with us at any time using our contact forms at echa.europa.eu/contact. We'll now have a 15-minute break, and then I'll hand over the floor to Ulla, who will be moderating the Q&A, and the experts who are here to reply to your questions. We'll start shortly.
of the webinar will be answered in our questions and answers document that will be published soon on the ECHA website. So if your question is not answered during the event, check the published questions and answers. Okay, let's get started. So let's start with some of the questions you have sent in advance. So there is a first question that on the Slido on the EUCLID. So thank you very much for this webinar. I would like to know if registrants are expected to attach models and predictions checklist to EUCLID. Who would like to, in the panel, take this one?
Yeah, maybe I can make a start and start this live and Q&A session. So hello again. That is really a good question because there is something new presented. So one can ask, so what is the obligation for me? Then I would like to go back to the roles. So the QSAR developer, they should fill in the QMRF. Then QSAR users should fill in the QSAR prediction reporting format. And from the regulatory side, so us at ECHA, we would then fill in the checklist and check if all the elements are there that are required under REACH. So this means we don't have really an expectation that the checklists are attached to the EUCLID by the registrants. I mean, of course, if somebody would like to go through and attach this, fine. And it may be very valuable actually to go through and to check if all the elements are presented in the EUCLID file, but there is not really a requirement to do that. So it will be us at ECHA to fill in the checklist and then to assess if the QSAR predictions, if used as a means to fill a data gap, if it fulfills all the requirements that are set out in REACH.
Thank you, Doris. Maybe a question related to your presentation as well, Doris. How should the checklists be filled for UVCBs or multi-constituent substances when there are many constituents?
Yes. The QSAR prediction assessment framework, I think it started with having a single constituent or a single substance in mind, but it can be used for a number of constituents. For example, if you have a UVCB or a multi-constituent substance, and therefore it was added to this checklist for QSAR results. Of course, when you think about UVCBs or multi-constituent substances with many constituents, it may be getting really cumbersome and maybe not needed to do for each and every constituent an own checklist. So now from the registrant side, what we expect is that for the constituents or representative constituents and impurities, if they are relevant, that there is an QPRF available and attached to the EUCLID, or an equivalent information is provided in the EUCLID dossier. And then on our side, we would check all this information with the result checklists, and then also assess the QSAR result based on this additional assessment element, and then come to a conclusion.
Thank you.
I think the question may be from like an authority side more. So how should the checklists be filled? So it's the authorities to fill, but again, it can be helpful for registrants to go through if they have addressed all the aspects that are relevant for us.
Okay, thank you, Doris. Then we have a few questions on AI. Just a second, I will. So there is a question on what is the ECHA view on current developments in using AI models to predict chemical properties?
Okay, maybe I can take this one. So, of course, we acknowledge that artificial intelligence is very dynamically developing field. We see a lot of applications, mainly in the knowledge discovery sector, but we start to see as well more and more QSARs or models which are trying to predict properties based on those AI algorithms, and to some extent it's nothing really new. We've been exposed to those models for last few years already, and we've been assessing them, and sometimes we've been accepting them. So there is no real difference whether the model prediction comes from the AI-based model or more traditional kind of regression model. We will assess them in the same way. So as long as documentation is clear that we can follow the logic how the predictions have been derived, and we can assess as well, how is the local performance of the model, how it performs on the closest analog, I think everything is fine. Of course, you may have a bit of doubt, what about this ambiguous algorithm requirement from the QSAR assessment framework? But in practice for us it means that we can reproduce the prediction. So basically, if we can run the tool, reproduce the prediction, that's fine for us. So that would be my answer. Thank you.
Thank you, Thomas. There was also a related question on the AI. Okay. Okay, it has moved. Anyway, we will come back to it. Yeah. Back to it later.
Sure, no
then the next question: when we update a registration dossier, are we supposed to revise QSAR predictions and fill in the additional fields in Euclid, or is it enough to do it for new dossiers, endpoint study records? And then related to that one, there is another question and as well, which is related to the updates, and it also has the follow-up question that is it necessary to update the QSARs in 6 months or 1 year time from today? So maybe one of you can answer updating the registration dossiers.
Okay, I can take it as well. So in principle, of course, whenever you're updating your registration dossier, you should check whether all the information which you have provided before are up to date and relevant. And of course, if you, after this seminar or after knowing the QSAR assessment framework, assessment elements from this framework, you will come to the conclusion that it would be nice to update your endpoint study record for QSAR to give us extra information to allow us basically to increase the chances that your prediction will be accepted, then it's fine. But if you think that everything is fine with the prediction and you don't need to do anything, you don't need to do anything. There is no additional requirements and the technical completeness check level that we will reject your dossier because you didn't fill in additional fields, especially those which Andrea mentioned in his presentation. Those fields will be introduced in the next release of Euclid to simplify your life for the future, to be more explicit on certain elements, but you don't need to fill them in if you didn't have them. So you can simply submit your dossier like it was, and it should go through the technical compliance check without any complications.
Thank you, Thomas. Next question. Could you provide a list of QSARs for which no QMRF is needed, as the information is publicly available? Who would like to take this one?
Maybe I can take this one. Yeah, we have seen in the past models where there is publicly available information, but not really a QMRF. And we do check if there is no QMRF attached. We can check if there is the information available that we need to assess a model. However, I would like to remind the registrants that it's in their responsibility and it's in their interest actually to provide the information, because situations can change. There can be a new version available, or the information is not anymore accessible to us publicly. And in this case, we then are in the situation that we don't have access to the information, and then we would go back to the registrant and basically send a decision to fill the data gap.
Thank you, Doris. There is also a related question to the QMRF. When QMRFs are assessed by authorities in the context of a registration, will the results be shared publicly? And is there an OECD effort to consolidate the different authorities' expert views? Who would like to take this question?
Hi. Yes, so I can take this question. Hi, everyone. So the challenge with the sharing assessments of QMRFs is that the assessment depends on the purpose of use of the model and its predictions. So we or OECD cannot publish a list or consolidate different authorities' expert views because they can legitimately differ if the purpose of use of the model is different. Furthermore, we have seen in the past misunderstandings that if a model is scientifically valid, then there was the expectation by registrants that all its predictions will be valid too. Now, of course, this is clarified with the QAF, with the framework that we check each prediction individually, but we can anticipate that would such a list of valid models be published, there could be the wrong expectation that every one of their predictions will also be valid. So unfortunately, at least right now, we are not in the position to provide the list or to think about a project like that.
Thank you, Andrea. Maybe you can continue with the next question related to your presentation as well. How to report in EUCLID QSAR results based on multiple predictions?
Yes. This I think we have seen a couple of similar questions on this topic, and indeed the concept of QSAR result as a combination of multiple predictions is new in the framework, and because of that we are still adjusting its reporting. So as of today, the best way to report a QSAR result is to fill in an endpoint study record, which reflects like the reasoning on how the different predictions are put together to get to the result. However, we know that there is the need to have a QSAR result reporting format to really give the possibility to structure information on QSAR results. And ECHA is leading, together with the EFSA, the Food Safety Authority, a project to develop such QSAR result reporting format. Based on this result reporting format, the same project will also give advice on how this could be further implemented in EUCLID. Right now, what we are looking at is the possibility to have individual endpoint study records for each prediction, and then one additional endpoint study record for the QSAR result. But this is right now work in progress, because as I said, the concept of QSAR result is new in the QAF, and we are looking at the best way to report it in EUCLID.
Thank you, Andrea. I think I keep you, Andrea, still for the next question related to the previous one. If you have anything to add, there is a question: how do you weight QSAR predictions and the QSAR results in dossier evaluation when you have multiple tests, outcomes, and results? Do you use only the QSAR results?
Okay, I think like this question may be interpreted in different ways. The way I understand this question is that the question is, I have multiple QSAR predictions. They may even disagree among each other, and then I have a QSAR result. What is ECHA going to check? Is the QSAR result or each of the individual predictions? If this is the meaning of the question, the answer is that we start by looking at the QSAR result. But of course, to verify that the QSAR result was correctly determined, we will also have to go back to the individual prediction. So we are going to check everything. And if there are different outcomes, different the predictions predict in different ways. Hopefully the QSAR result justification explains why the conclusion goes into a certain direction. Maybe one of the two predictions was completely unreliable and out of domain, so only the other one is taken into account, or similar. So that's how I understood the question. That would be my answer to the question. In case that was not the meaning, of course, please follow up in slide and then we can adjust the answer.
Thank you. Next question here is related to the similarity context or concept. So similarity is a broad concept, can be calculated with different methods and considering different structural features. Are there any plans for harmonization or for giving advice on the similarity? Who would like to take this one?
I could take this one. Indeed, it's a really good question, and there is probably not a really easy answer or not a straightforward answer to that. It depends a little bit. I think it depends also on the endpoint, what we do normally in practice, we look a lot on functional groups that should be presented in these similar substances. Maybe not so much at, for example, Tanimoto scores. That is also one possibility to address similarity. But with these Tanimoto scores, you risk that you miss out a specific part of the molecule that may be related to the property. So what we tend to look is that functional groups are covered in the similar substances and those functional groups that are also related to the endpoint. Maybe to add there that it's not necessarily limited to look into the training set. When you look for similar substances, they can also come from other sources, for example, the OECD QSAR toolbox. So it's a really good source to look for analogs. And if I may advertise a bit, the OECD QSAR toolbox, it has also a lot of mechanistic profiler where you can really use it to find good analogs that are mechanistically related. So I think it's really worth to check also via the OECD QSAR toolbox. And this is, I think it's really an important aspect when we assess a QSAR prediction, if it's reliable and adequate. So we pay a lot of attention on similar substances and if they are predicted well by the QSAR model.
Yeah, I think I could add that basically there is no need to focus on the mathematical methodology to find these analogs. When we look at the analogs, sometimes we are presented with the Tanimoto coefficient, and for an analog it's 0.9 and for the other one it's 0.85. We don't do anything with this number. It's not that we say, okay, it's below 0.8, we don't consider this a good analog. If above 0.8, we consider it a good analog. No, we don't do that. What we do, we look with our expertise on the endpoints if the analogs make sense, if they can really provide information on how good is the prediction for the new substance. If from endpoint point of view these analogs make sense, they are good for us, irrespective of the Tanimoto coefficient, the die score, whatever algorithm is used to measure similarity. So please do not focus on the mathematical methodology, focus on the endpoint and mechanism and like what makes sense for an expert.
Thank you. Maybe we can continue a little bit on the assessment side. There is a question on when are you going to start using assessment framework for compliance checks? So, Thomas?
Yeah, I can take it. Yes, I can take this. Okay, I think as it was explained during the webinar, assessment framework from our practice point of view is not the novelty. Basically, what we've been trying to do, we've been trying to share our experience from 10 years of evaluating QSAR results within REACH with the OECD community, and based on that build the assessment framework. So our practices are not really changing. What is changing is what may change in the future is a bit how we are referring to the different legal arguments in our decisions. So this probably will happen from the next year, but it basically is not changing our practices, and only change which you may see is, that we will, if we will start to refer to the assessment framework principle in our decisions, then we will as well update the ECHA guidance on QSARs and grouping, and basically will make the reference to the QSAR assessment framework there, so that you have a consistent legal basis for our decision. But that's more kind of legal support information rather than the practice. So I think from your perspective, nothing really will change, and I wouldn't worry about it at all.
Maybe to clarify on guidance update. So guidance update is a very long process which involves member states, etc. Right now, this is not what ECHA plans. The plan is eventually next year, when we start referring to the framework in compliance checks, is to add just an introductory note to the ECHA guidance on QSARs. This is Chapter R.6 of the guidance, and say that in our assessment, we will use as legal reference also the OECD QSAR Assessment Framework, and that's it. Any other plan for bigger update is not at the moment on the table, because we put a lot of work and effort in having the OECD QSAR Assessment Framework, and we don't want to duplicate the work now in a guidance update. But we will find ways to have clear instructions and explanation of what we do and how we use the framework in our practice. And today, 1 year before we really start to do this, we have already started the communication and the explanation.
Thank you, Andrea and Thomas. Maybe a follow-up question on this. Are there new EUCLID fields for the QSARs parts of the TCC already now, or when are they implemented in REACH IT as business rule failure? Would Thomas or Andrea, either of you, want to follow up on this? Or
yeah, I can. And the new fields, which will be available in 1 month or so, will not be part of the technical completeness check, at least not now. In future, we may change this, but we are not going to do anything that is like too heavy from registrant side. What we started to discuss is whether the field applicability domain, which is a pick list, yes, no, borderline, will be made available. So for now, no changes to the technical competence check. Maybe in future, if we do, it's going to be a light one, not every single field to be provided all the time.
thank you. The next question, let's see. Can you confirm that being outside the applicability domain for certain descriptors does not mean that the model is unacceptable, if it can be discussed as acceptable considering the other descriptors that define the domain for prediction model? Anyone with applicability domain?
Yeah, I think I can take this one. So I think the short answer is a yes, we can confirm that. And the whole point of the QSAR assessment framework is to list what is important, but also to leave then the flexibility to assessors to focus on what matters. And I mean, of course, when you have a parametric range, you can be just a little bit outside this range, and therefore being out of applicability domain formally. On the other hand, if we can see that there are very good analogs in the training set that are well predicted by the model, and everything else seems to be fine in the prediction, we are not going to fail the prediction only because formally it happens that for one parameter it is outside the applicability domain. However, if we see other issues in addition to the applicability domain, most likely we are going to list all the issues that we found. And among these issues that we have found, we may also pick on the fact that formally the substance is outside applicability domain. This is more because of internal practice. Once we find incompliance, we better list everything. But that doesn't mean that if that is the only problem, we are going to send a decision just because of that.
Thank you, Andrea. Maybe a follow-up question, Andrea, for you on this. Is there a plan for harmonizing the applicability domain definition of models, or at least to set some minimum requirements for applicability domain definition?
Okay. Short answer, no, or at least no by ECHA. And this was discussed during the QSAR Assessment Framework meetings of the expert group. And the challenge we had there was that the QSAR Assessment Framework was a framework discussed among regulators, while for the definition of applicability domain, I mean, of course, we needed the counterpart of model developers. However, there has not been agreement in many years on how to best define applicability domain. And I'm quite satisfied of the pragmatic solution that was found in the QSAR Assessment Framework. That is, the model developers, hopefully they know what they are doing, and we are going to trust whatever definition of applicability domain they give. That's what we check in the framework. On the other hand, we know that there are some aspects that are very important to assess the validity of a prediction, irrespective of what is the definition of applicability domain given by the model developers. And all these aspects is what we have gathered under reliability of the prediction. So another way to look at this is that the assessment elements that we have under the reliability of the prediction is kind of applicability domain for the specific context of use for that specific prediction. But when it comes to the formality and of defining applicability domain, there was no agreement in the scientific community. So regulators, I think they cannot impose that. It should be the scientific community coming with the definition, then we will use it. In absence of that, we found a solution in the framework, which I think allows everyone to do the work of evaluation of QSARs.
Thank you, Andrea. Next question is if you have related to the availability of multiple models for one endpoint. So the question is, do all available models for a specific endpoint need to be used and then combined to a result, provided they are applicable and give reliable results? When is it enough to use one specific model to fill a data gap? Doris, would you like to take this one?
I can take this one, thank you. So thanks for the question. Maybe to start with, it's in a way up to the registrant, and it's also the obligation by the registrant to provide adequate QSAR results or QSAR predictions when they want to fill a data gap. There is certain conditions in the REACH mentioned in the Annex 11. And so we don't expect that each and every model is used that is available to provide a result or prediction. However, it may be in the interest of the registrant to check more than one model and to see if they are in agreement. Because if there are discrepancies, I mean, it's better for the registrant to understand why, and if one of the models don't predict well a substance property. So I think it's good to check some more models if available and if it's easy enough. Also, maybe depending on the substance and the complexity of the endpoint, you may want to look a bit closer for substances that are more tricky to predict. Maybe if you have a straightforward chemistry, maybe you are satisfied with one model. And then it has to be also justified when more models are used and that is combined to a result. It has to be justified how this is done, and there is one assessment element then to address this. So overall, no need to overdo it maybe, but still be careful and have a look if the chemistry requires maybe a more cautious approach, and maybe you need to address the substance with more than one model.
Thank you, Doris. Maybe we can follow up a little bit on the related to the models. So there is a question: what kind of data for models development do you consider reliable? Do data must be obtained from OECD test guidelines? If yes, it may limit the possibilities to develop new acceptable models, and it would slow the shift to non-animal approaches. Would you, Thomas, like to follow up on this?
Okay, yes, that's a bit tricky question, but I will try to answer, and then I will ask colleagues if they want to complement. So one of the the new element for the prediction is the fit for regulatory purpose, and of course, in general, answer is yes. You could do whatever, you could use whatever type of data to build your models. But then, when we are talking about rich context, and when we are talking about using QSAR predictions as an adaptations to the standard information requirements in the rich registration dossier, then we are talking about replacing or adopting concrete studies like Ames mutagenicity, short aquatic toxicity to fish, short aquatic toxicity to Daphnia, skin sensitization. And those under REACH, they are very prescribed what it means, standard information requirements, and normally there is a standard test guideline or test guidelines associated with these requirements. So from our perspective, it's very important to make sure that the data, at least majority of the data which have been used to build a model, is indeed derived from this kind of studies, because otherwise we really don't know whether the property which you are trying to predict is fully in line with our standard information requirements, and then we may have a problem with this fit for purpose criterion in the REACH context. So I would say, in the context of REACH, I think that's more or less the case, that we would like to see the training sets compiled from the guidance studies which are meant to cover these information requirements, if you are trying to use the QSAR to cover it.
Yeah, maybe what I can add is that, I mean, of course, what Thomas is mentioning is the ideal case, and we know that if we put this as a requirement for the models, there is not going to be many models that will be accepted. So keeping like as the ideal case, what Thomas just described, we are also looking into ways to be able to accept predictions when we don't have, let's call it, the perfect training set, or it could even be the perfect training set, but we may not have access to the data to check that this is really as good as it said to be, etc. So one way that we found to deal with this situation is to at least look at the data for the closest analogs. So if we don't have information on all the data in the training set, or even if all the data in the training set are not perfect, we would like at least the data for the close analogs to cover the key aspects. And one example is a problem that we sometimes have is predictions for bacterial mutagenicity. That means the fifth strain from Ames test. Now, luckily, many models started to be explicit about the fact that all 5 strains are covered. But if they are not explicit on whether the fifth strain is covered or not, but they present analogs and they provide reference to the data for the analogs, if we see that the analogs have the fifth strain covered, that could be good enough. Another aspect that we look at when investigating the quality of the data used to build the model is also, we know that in a QSAR model you may have many data points that may fit the trend. So what can happen is that you have a trend, you know that some points in the trend are good enough in terms of quality, and for some others you don't know. However, if these other points for which it's unclear how good quality they are, if they fit the trend of the good quality points, then we start to be more confident that the whole training set can be used to generate models and therefore predictions that are good enough. So yes, the idea requirement is very high. We want to promote the use of in silico methods, and we try as much as possible to use this like other possibilities when we are not looking at the ideal case.
Thank you. Maybe we continue with the models. So there is a question on, will you provide best practice examples on assessment framework for the most frequently used QSAR models? If not, may you explain why you came to this decision?
Okay, I can reply to this one. So first of all, OECD, after publishing the framework, has published also several examples on how to use it, and these examples were prepared by other authorities outside Europe, and of course there was a review process where also other experts, including those from ECHA, looked into those examples. Nevertheless, right now we don't have any rich example, and we don't plan to have a rich example in the near future. The reason for that is that it takes a lot of time to prepare the example, and we are right now in the process of finalizing our internal policies and workflows to use the framework in our assessment. It could be that 1 year from now, when we start to use the framework in ComplianceSec, we could be in the position to publish some of those rules, even the workflows we plan to follow. And example, the challenge with example is that with REACH, the responsibility of the assessment lies with the registrant. And the challenge we constantly have when we provide an example is that if we provide an example and we make a statement about the property of a substance, there could be the perception that we are taking this responsibility on us, on how the substance behaves or is predicted to behave, and taking it away from the registrant. And that's very tricky for many reasons, including legal ones. So that's always difficult for us to build a like precise example. Nevertheless, let's see 1 year from now, if we are in the position to give more instructions and perhaps examples. I think we'll do our best to facilitate everyone's life, because once again, when registrants do a better job, it's also our job is easier, and I would say even more rewarding. So it's in our interest too.
Thank you, Andrea. The next question is related to robust study summaries. So is it still requested to provide a robust study summary for the respective endpoint for each of the read-across substances used in the QSAR results? Who would like to take this one?
Maybe. Yeah, I understand when it says here read-across substances, it means the similar substances to support the QSAR predictions. And in this case, the answer is no. So we don't expect an own robust study summary for a similar substance. However, in order for us to be able to assess as a prediction and the similar substance and the performance of the similar substances with the applied model, we need to have some access to those similar substances, to the identity, and also to the experimental data. So I think that's the essence. So we need to have enough information.
And I think this links to another similar question that we haven't answered yet. That was about dossiers that are part of categories, and then whether the information on analogs needs to be provided twice. I remember seeing it popping up at some point. So I think it's a good time to answer that, and then we get rid of it when we find it in Slido. And so the new fields, because the question was referring to the new fields in Euclid to report about the analogs, that table that you will be able to compile is quite light. There is one field for the identifier that will be the smiles or the cast or whatever works best for you. one field for the experimental value, one field for the predicted value, one field for comment and I think also one or two fields for the reference of where the study comes from. So it's a relatively light table. So if you have a category of substances and you have multiple dossiers, yes, you have to repeat to very limited extent the information, but you can be reassured that we are not asking you every time to attach new reference substances and create new entities and lose a lot of time. If you have in mind what modern QSAR tools provide you as information on analogs, that's pretty much what we will like, but you will have the possibility to indicate in EUCLID.
Okay, thank you, Andrea. Let's see when the question pops up on my view, but there is a related question. I think this is not exactly what you were referring to, but the question is there a plan for a new EUCLID endpoint study record similar to the weight of evidence justification for summarizing multiple predictions in the QSAR results? So would you, Andrea, like to?
Yes, I think this was partially answered by me before in the context of how to report in EUCLID the results. And let's come back to this because this is a new concept and it's important. So right now, as I explained, registrants need to fill the information on the result as endpoint study record. However, we don't have yet a result reporting format or any dedicated field in EUCLID to report the result. So, of course, the fields of the conclusions, results, remarks, everything can be explained there. But ECHA, together with EFSA, is now working on a better reporting format for results, which could be also reflected in EUCLID. So, although there is not ideal solution now, and now I think registrants will have to be creative as they have been in the past to summarize the information in summaries in the same endpoint study record where the prediction is given. In future, this information might be more structured in a way that is either similar to the weight of evidence, as the question was hinting, or the other thing we are evaluating is similar to a read-across. In read-across, we expect to see source studies, experimental studies separately, and then one endpoint study record to justify the read-across. The option we are investigating for QSAR is also that to have individual endpoint study records for each prediction, and then one new endpoint study record, the QSAR result, just to provide the justification on how these results are put together, how the predictions are put together into one result.
Thank you, Andrea. Then we have a question related to AI. I think we already had one, but let's see if there is something new on this one. So thank you for this webinar. AI is very much in the news. Are there any plans to review and assess the potential use of AI in predicting toxicity? I think, Thomas, you replied before on the AI, so do you want to?
Yes.
Continue?
So apparently this is our missing AI question, which we've been looking before. Yes, so I think I partially already covered that. So in principle, we don't see any, when we are assessing the predictions, we will not discriminate whether the prediction is from the AI-based model or the more traditional model. And then maybe more explicitly about predicting toxicity. When you are building the model, you have always those two elements. First is how to compile extensive, robust, and reliable training and validation set. And then the second part is, how to associate different features from structural features to predict the properties and to demonstrate that you have indeed. AI can help in both of this, because we can see that more and more that AI algorithms may look for the new data, can already pre-assess to some extent quality of this data, and can start to build almost kind of simultaneously the training and validation sets. And then as well, when we are talking about finding the relationship between different structural features and the predicted property, then this non-parametric approaches which AI offers may be as well very powerful, and it might give you the good results.
and we probably will see more and more those models in the future. But as I said before, as long as it is well explained how the data were identified, what were the curation protocols, basically what kind of quality assurance that the developers took into account to make sure that the data which were used to build the model was of good quality, and what kind of algorithm were used to find this relationship between properties and the structural features, then it's fine. And and anyway you have the performance will check how the model perform in the local chemical space, how the close analogs are predicted, whether the close analogs are close enough. So from our perspective, there is no big difference, I think. So yeah.
And maybe it's also a matter of how AI is defined, because today everyone likes to talk about AI, and 10 years ago I remember going to conferences and being asked, do you accept prediction from neural networks because they are black box? Now they just replace the word neural network with AI. Some people have this interpretation of AI, and in this sense, I mean, that's a model absolutely as any other. We would probably not even call it AI if it's just a neural network with a classical training set predicting a classical endpoint. Also a story, we had a couple of months ago a training on AI in toxicology in ECHA, and then we go there and then they tell us how to build QSAR models, and then there was the perception that, okay, maybe we have a different understanding of what AI is. There is a lot of discussion on what maybe is another type of AI that is now being developed, AI that reads hundreds of documents, and from those documents understands if something is toxic or not. But that is not QSAR, that is not discussed today. There is no answer, I believe, in ECHA or probably anywhere on what to do with this type of summaries or results that can come out of such type of AI. But if by AI we mean the neural network, artificial neural networks, or similar method to build QSAR models, for us, really, as Tomasz said, there is absolutely no difference. They were around 10 years ago. We were evaluating them 10 years ago, accepting them sometimes. We will keep doing the same today.
Yeah.
What I see is maybe a potential issue is the data quality behind. So if a model searches in the internet for data, we don't know the quality, so this is really difficult to assess them for us. So we need to be sure that the input for the model is also adequate for our purpose, and then only then we can use the output, meaning the prediction.
Yeah. Therefore, it's very important to really report how quality assurance, because of course you can use those AI algorithms to identify additional data points, but then somebody, or at least you need to put clear filtering criteria on which under how you will basically narrow down those information and make them available for the model. So you need to apply some kind of quality criteria. And just to maybe finish with so if by AI you are thinking about ChatGPT, so please don't submit as the ChatGPT prompts whether the substance is toxic for reproduction. First, because most probably ChatGPT will hallucinate a bit about it. Second, because tomorrow you may get completely different answers. So all idea about reproducibility is completely gone. So if your question was about whether I can use ChatGPT, then my answer is not yet. No. Thank you.
Okay, thank you. I can see a few questions related to QSAR Toolbox, so maybe we can go through those next. So the first one is, will the QSAR Toolbox create the QPRF in the new format, including the new sections? Andrea, would you like to take this?
Yes, and I think indeed that this links also to the other question we have seen before, is the QSAR Toolbox compliant with the QAF, etc., because it's developed by ECHA, etc. So we update QSAR Toolbox once a year. There is one public release every year, and the next public release will be at the beginning of June. I think we are at version 4.7. This release will include the updated QPRF, the updated QMRF, and interestingly also the justification document from the QAF that will present assessment elements so that the users can reflect on those when justifying manually a prediction. I think it's very important for every QSAR users to realize that most of the times that the QSAR toolbox is used under REACH, this is done for read across. And we have clarified in separate webinars that if you are using QSAR toolbox to prepare a read across, including trend analysis with few analogs, that is a read across with a trend in potency, please refer to the read across assessment framework and not to the QSAR assessment framework. We understand that there could be confusion because it's called QSAR toolbox, but if you're doing a read across with a QSAR toolbox, that's a read across. And we have rejected, I think, many studies where the read acrosses from toolbox were presented as QSAR models, and that didn't work at all. So this is in general about QSAR toolbox, QAF, etc. So for the specification, we'll create the new QPRF, yes, but when you generate a QSAR prediction. And the QSAR prediction can be generated in toolbox by running external QSAR models. I think it's also good to remind that a model that is in toolbox doesn't mean that it's endorsed by ECHA, doesn't mean that the QPRF it will produce will be compliant and the prediction will be compliant, but the fields will be there, and I think many of them will be to be manually compiled by the users when the toolbox cannot do it automatically. But at least the user is presented with the fields and has a chance to reflect on those and then decide whether to go submit the prediction, or if there are like points that cannot be addressed, maybe could be better to do something else than just using that prediction.
Thank you, Andrea. So I think we covered the question also related to assessment framework and relationship to the toolbox on the same answer. Thank you. So the next question is how availability of data may affect the external validation, and is it mandatory for regulatory acceptance? So for example, in cases the software is proprietary and training sets are not available, or there is protected data in commercial software. Anyone would like to take this?
I can take this one. Yes, so external validation is in principle very important to check. And it is a mean to check if the model is overfitted or not, meaning if you have very small training set and a lot of parameter, it may work well for your training set data, but maybe it's not very predictive then if you predict a new substance. So this is a really good check. However, in some cases, it will be difficult because maybe you have small training sets already, so you cannot truly split then and then address this external check. And in some cases, if they are mechanistically built, well built, we can be quite flexible on this, I would say, on this external validation. So for us, I think the local performance is much more important than because we assess individual predictions. So maybe it's different if you have a screening of a large database, then you want to know that overall you have a good output and the predictions are overall reliable. But we look at individual predictions, so local performance is, I think, more important for us. And so that's why we emphasize already many times that we look at performance on similar substances. And maybe something that links back to the previous question on the QSAR toolbox, because our QSAR models were mentioned. We do have in the QSAR toolbox the possibility to build via the trend analysis local models, and there we don't have external validation. However, if you think of a model built on, for example, 10 different alcohols, so they are maybe it's a homologous series, so I think it's not so important that you have an external validation in these cases, but you get quite confident if you have an interpolation, your substance is somewhere in between, and you have around the same type of substances, that's good enough for us.
Yeah, and maybe to be even more explicit on when external validation is needed. As Doris said, I mean, mode of action, understanding of mode of action, good analogs, they are important, and especially, I mean, this is possible to do when we are facing a mechanistic models. If we are looking at a statistical model where the only information we have about how good it is are its statistical performance, in that case, it becomes important also the external validation, because the external validation is the only indication that the model really works. It's different from a mechanistic model where we have a mechanistic understanding, we have a new substance that we want to assess if it can be well predicted by the model. We understand if its mode of action is covered, if there are analogs in the model training set that have the same mode of action that are well predicted. And if they are, the external validation in that case may not be required.
Thank you. I think I continue with a question related to performance of the models. So what is the level of performance of a QSAR model ECHA is expecting to accept a QSAR prediction?
Maybe I can add on this one. It's indeed linked with the previous one. I think it depends on the purpose, basically. So it's difficult to really have a value. There is in the QSAR assessment framework, there are some values on the performance predictivity. There's R square and I think Q square given as examples. But overall, it will depend on how close the prediction is to a regulatory threshold, like to a criterion, and how certain you have to be. Yeah, that would be my answer to this question.
Thank you. Let's get back to the models. So the next question, expert models such as direct nexus do not have a clearly defined applicability domain. So how reliable are negative predictions in those models? Who would like to take this?
Okay, I can try to start, and maybe then if somebody wants to complement. Okay, so first of all, direct nexus per se is not QSAR model, is the structural alert system. And the difference here is that we are not, that it does not provide you the quantitative predictions for the property based on the structural features of your substance. It identifies certain structural features which are associated by expert and within the knowledge base with some specific toxicological effects. So it is indeed more expert system to identify whether you could see some concern within the substance. So in this, from this point of view, of course, you cannot have an applicability domain per se, because it's a basically identification of the so-called structural alerts. So we are not, we don't believe that the alert as a in standalone can be used to cover information requirements. However, there are some more specific. LASA is developing more specific models for specific endpoints. I think they have a model for mutagenicity, for skin sensitization. When then all the principles are covered, you have a training set, you have a prediction, and then all principles are there. But don't think it's under the same platform. But this is not direct nexus. So that's just to differentiate. And by the way, this confusion as well is very often done for the QSAR toolbox. QSAR toolbox profilers as well are the structural alerts, which are tools to find the analogs, which have a chance to behave in a very similar way, to build then your prediction justified, not to rely on those outcomes from those profilers and treat them as a ultimate prediction of your property. So that's the difference here. And regarding the negative prediction, so even more, even if you, because you could still say, okay, but we could, if we don't see any alerts from Derek, can we assume that there is no problem with this substance? This could be the case if we have a certainty that for given endpoint, all possible type of toxicity have been covered within the Derek alerts. But because this is not the case, because alerts are developed and covering only the part of the knowledge which we have and understanding about endpoint which we have, and does not cover unknowns, I think it's very difficult to basically justify the negative prediction solely based on the Derek alert. Of course, you could use it as a supporting evidence for read across, for example, and we are using sometimes Derek alerts just to substantiate, for example, some group, and basically to demonstrate some similarity in certain properties, but this is then not unique line of evidence which we are using for doing that.
And maybe, I mean, if we agree to what Tomasz has said, just so to be precise with the terminology, the question asks how reliable are the negative predictions. So as Tomasz mentioned, the in vitro mutagenicity and the skin sense models, I think they explicitly call the negative results as negative predictions, and there I believe there is information on analogs, perhaps even on the training set, and that's okay. The question did not mention explicitly whether the intention was to use lack of alert as negative predictions. If that's the case, for the reason explained by Tomasz, please don't do that. Also remember that direct nexus often addresses high-tier, very complex endpoints, and we have explained several times in different webinars why we are not confident in using in silico tools for those endpoints. But they can be used to substantiate read across and for other similar type of investigation.
Thank you. Let's move on to biodegradation simulation studies. So the next question is biodegradation simulation studies on UVCPs or multi-constituent substances are technically very challenging, but QSAR models that produce actual half-lives and metabolite formation rates are scarce. What combination of QSAR models and types of output could be accepted to cover this endpoint? Doris, maybe you
I can
would like
take
to
this
take
one,
this.
yeah. I mean, a lot is already, I think, in the question itself. I mean, it's all true. Biodegradation simulation studies are really difficult to conduct for certain substances and difficult to interpret. There is not a lot of data yet available overall when we think of, like, we have under each 30,000 substances registered. So it's not yet so well covered. And QSAR models we have more for already biodegradability. Some try to address also biotransformation pathways and also to try to address simulation studies. It's still difficult, I would think, to reflect the real biodegradation simulation study outcome by QSAR models, though they can be really useful, especially for UVCPs or multi-constituent substances when you think you don't even know where to start to test. So those models can really supporting testing strategies. For example, to highlight potential candidates that may then be PBT substances, so having persistence, bioaccumulative and toxic properties. Or now we have the new hazard class PMTs and VPVMs, so persistent, mobile and toxic substances. So in this case, models I think can be really valuable to identify where to look closer. And then also to see what potential degradation products could we expect, and to look for those. Maybe also to do literature search on that. I mean, it's a bit difficult now to say what would be sufficient. Normally, when you use such data, it's always in a weight of evidence. So anything that would come from a QSAR model or a prediction would be then embedded in a broader weight of evidence approach together with other information.
And Doris, I will try to be even more explicit, but please correct me if that's not the case. And I would like to be explicit on 2 points. One is something that we do not accept, although we see it several times, and maybe this is a good platform to say this is not good enough, is when the models predict half-lives, days for the half-life, based on training sets for ready biodegradability studies or BOD. We don't accept such type of models, otherwise the requirement will not be there. There will just be BOD, and then there will be a calculation and extrapolation to find half-life from that. So that is not good enough. And yeah, I would also add that as of today, to my mind, it doesn't come any single model that gives predictions of half-lives that we would accept as they are. We have looked into several models, hoping to find at least one where the error was not too big, with training sets good enough in terms of quality, big enough in terms of number of substances. And at best of my knowledge, there is not such model as of today. The other way to say it is to use whatever you have as a weight of evidence, but to be more explicit on how to use QSARs for this case specifically, I don't think we have QSARs that are good enough for the half-lives. I think it's much better news for the identification of degradation products, as Doris was mentioning, because there is a number of cases where, for example, the substance rapidly biodegrades, and then you don't have to test the substance for toxicity, but some of the degradation products and similar type of activities. In that case, we like the predicted degradation products to identify further testing needs. I think in many cases they have been considered good enough. So at least in this sense, it's better news.
Okay, thank you. Related to this question, let's stay on the environment. More practical examples of common models, for example, BioWin and ECHA, would be helpful, or are such examples already available? And I guess this also relates to other than environment endpoints in general, to the models. Would you, Doris, like to?
Yeah, I think Andrea had already one question on examples, a bit similar. I mean, we do have, especially BioWin, ECHA, they are so long around. They are already presented in the guidance document from ECHA on the information requirements and chemical safety assessment. It's a developing field. New models are popping up nowadays, even like faster with the new technology. We will see how to implement the QAF in our internal processes, and then see if we can be helpful also for registrants. Not sure if we will, how far we will get with examples. Maybe I give back to Andrea if he has a different view on that one.
I think that the best information you can find from ECHA on this is from the webinar, the ECHA webinar from 2 years ago that I mentioned at the beginning of my presentation. I was now quickly checking the slides. I mean, they are not full examples on BioWin and QSAR, but we take different screenshots to present different aspects on how we assess those. It's not the example as probably we would like to have the full example, but it's the best we have at the moment. Today, there is no point for us to make an example that is not following the framework, the QSAR assessment framework, because it will be outdated soon. So linking back to the previous reply on examples, we'll see what we can do. But if you keep in mind that in ECHA we will start next year to use this, then I think that's the timeline you should expect also for the examples.
Just maybe just a follow-up question. So if we already have the examples existing, they are still applicable, even are they still applicable for the assessment framework as well?
Yeah, in a slightly different way. For example, in our current practice, we check the parametric domain under applicability domain, while the QAF tells it to check it under reliability, this type of things. But in practice, they are still true.
Good, thank you. Okay, next question. What is the correct approach when no QMRF is provided by the model developer or available online? Would that be a knockout criterion to use the prediction in general?
And
Andrea?
this
Yes.
is maybe easy question. Yeah, I think that the easy answer is use a different model. For most endpoints, there are many models available, and the documentation about the model is such a basic information to proceed with the assessment. Now, I mean, no QMRF, if the information is fully provided just in a different format, that's no problem. But if we don't have information about the model, then just use another model, because for regulatory use, that's not gonna work, at least not for ECHA.
Thank you. Related to the knockout criteria. The next question we have is the framework going to be accepting the results of Danish QSAR? Would you, Andrea, like to continue?
On the knocking out. I mean, Danish QSAR, I think the colleagues from DTU, they have two different platforms. One is the Danish QSAR database, probably is the most well-known because it has been around for longer. And then there are the Danish QSAR models. For the Danish QSAR database, that's a database of predictions. And in terms of applicability domain, they only have the information whether the prediction is in domain or not, without explaining why. No means to evaluate the reliability of that call, etc. So we usually don't accept predictions from the Danish QSAR database. It's a different story for the Danish QSAR models because those models are those developed by DTU and they provide many more details on the prediction. And for predictions from the Danish QSAR models, if everything we need from the QSAR assessment framework is provided, then they have the same chances as any other model to be and predictions to be accepted.
Okay, thank you, Andrea. Let's continue on NAMs. So there is a question, how are NAMs defined by ECHA? New approach methodologies, non-animal methods or other? Do NAMs still include a necessity for animal testing at certain points?
Okay, maybe I will take it. Yeah, and that's the general problem. If you are introducing new term without being clearly defined what the terms means, then what happens is that you are ending up with all those interpretation. And this is our fault because indeed in 2016, when we had the first new approach methodologies workshop, we introduced this term without being explicit what it means. And now we have to deal with it. So once again, for us, NAMs is not non-animal methods, is new approach methodologies. And what we wanted at that time, what was the idea behind the new approach methodologies is that we need to find a new way of characterizing the hazard. We need to switch from, traditional testing when we are basically trying to identify adverse outcomes. And based on those observed adverse outcome, build the whole regulatory system to start to look at the more subtle changes at the molecular level and try to start to build the regulatory system on it. That was the original intention behind new approach methodologies. And of course now, since the discussion is progressing, we would say we still believe that, so it is in the intention and in principle, it is looked at as an alternative to traditional animal testing. And these alternatives might be via different ways, via in silico methods, via the in vitro methods, via combination of different techniques. But and as well, but we believe that there is still, for some endpoints, probably we cannot do this step without some intermediate solutions. And those intermediate solutions is, for example, to start to use the new way, generate new type of information, like, for example, transcriptomics responses or metabolomic responses on the animal study, just to better understand the relationship between adverse outcome, which you will see in the animal study, and the level of changes at the molecular level, which you can measure. And by doing that in the more routinely, you will better understand what is this relation, which will lead in the future to basically to the situation that you may not eventually need at all do the tests on animals. So I would say that NAMs is more the tool, the way to achieve the goal on non-reliance on animal testing, not per se as a non-animal testing method. And that would be my answer. Yeah, thank you.
Thank you, Thomas. So then we have a question related to toolbox. I'm not sure, Andrea, maybe you already covered this, but just checking if there is anything to add. So do all QSARs in the ECHA OECD QSAR toolbox have the QMRF version 2.1 as background and thus could be used under REACH? Also, do these models have the QPRF 2.0 already?
I think there is a little bit to add. I said that yes, so the new toolbox will have QMRF 2.1 and QPRF 2.0. Now, how come that we have QMRF 2.1 in toolbox if it was just out? Did everyone update their model immediately? No. The trick is that to prevent trouble for the model developers in updating the QMRF, the only change from 2.0 to 2.1 in terms of QMRF is a new field called abstract, and it has numbered with a 0, so it doesn't even change the number of the other fields. Where modelers who want, they can add some more information on the model, like write a story about it. That's why there is this abstract field, but otherwise the QMRF is the same. We have updated the help text on how to fill each field of the QMRF, but that's it. So basically they had the QMRF 2.0, we added the abstract field, and I suppose it will be empty if that has not been provided by modelers. So, but that I think is not so important. More important is the major change of the QPRF. And to go back to what I said before, yes, predictions will come out with the QPRF 2.0, but remember that more often you have a read across and not a QSAR prediction with toolbox. But if it's a QSAR prediction, it will come with a QPRF. Unfortunately, many fields need to be filled in manually because they require expert judgment, and they will come out empty from toolbox, and then it's the user that has to fill those in.
Thanks, Andrea. Then another question that might be already covered, but related to the performance of the models. Can you please elaborate how you determine that the model predicts well with similar substances? Doris,
I can
take
this
take?
one. The prediction of the property, it's not a standalone information, but it's here for a purpose. So what we want to achieve is safe use of chemicals, safe for human health, safe for the environment, which means that any prediction has a regulatory purpose, and then prediction of similar substances would be seen in this slide. Meaning, if I have a regulatory criterion, for example, BCF of 2000, if I have now similar substances and they are all well below the criterion of BCF 2000, and my substance is also predicted well below 2000, then it's a very good indication that there is no concern. But if now the predicted similar substances are around the criterion, my substance is also quite close with the prediction, close to the criterion, then you need to be extra careful. So I think it's context dependent how close the predictions of similar substances should be. It should give the regulators confidence that the model performs well in this local chemical space, basically. That's the purpose.
And
Thank you, Doris.
I can add something in relation to a similar question that we just got in, so to also simplify a bit Ulla's life. And yeah, I see that Ulla, now you have highlighted the question. So the question was, if I want to show that the QSAR works well and I need to refer to analogs, do I need the rights to use those data? And for QSARs, you do not, because the idea is that when we look at the performance for analogs, we don't go all the way to check the robust study summary independently to check that the result was good enough even for a compliance check as a standalone. And when the user of data only takes the numerical outcome and maybe few information such as the test guideline, then there is no need to have any rights to use those data.
Good. Thank you, Andrea. Then the next question, with regard to the use of an unambiguous algorithm, many commercial software programs use their own algorithms and are not explained in detail. Are not used are not explained in detail, such as using BIML or AutoDoc. Is it enough to show what docking procedures were undertaken via known methodology, but algorithm is not included? Who would like to take?
I can take it. I think there are like a number of things that could be said, but also to stay short. So first of all, we don't see many docking models in REACH. I don't recall seeing any case in 10 years. I don't know about colleagues docking. I think for us, the point is that somehow we must be able to understand what's happening. The challenge I could see in the case described in the question is that we don't have access to the tool. We don't have access to the algorithm. So we kind of would have to trust what is given to us as a number without possibility to check the algorithm or to reproduce the result. That is going to be difficult if it ever happens. Again, I've not seen under REACH such type of models yet, but ideally it would be nice if the algorithm is not disclosed, at least to make us, to enable us to reproduce the prediction, to see that what is being reported is not just the result that is convenient for the assessment, but is the result that is given by the model. And I think because we don't see those models a lot under REACH, maybe it's best to focus on other answers. And maybe let me tell you a couple of things. So Ulla, I'm going to steal your role for a second, but I see information from our communication supporters that they want like to remind participants to answer to the live poll questions before leaving. And something else, we will close in 13 minutes. I'm not sure we will go through all the questions. So what I can advise participants is to vote for the questions they would like to be answered live, because I'm not sure we will manage to go through all questions. And Ulla, sorry for stealing you the role for a minute.
Yeah.
But
no worries. Thank you, Andrea, for noticing that. Yes, indeed. Answer the poll. Poll, you can find it under the live poll. Okay, let's continue with the questions. We still have about 10 minutes or a little bit less. So the next question on usefulness of the seminars. So there is these seminars are very insightful. Thank you. Is it possible to produce some detailed webinars on specific QSAR models? For example, how they were developed, the dataset selected, and also including the statistics and comparing the predictions to actual experimental results. Would you, Thomas or Andrea?
I mean, again, many things that could be said, but one thing that we are going to experiment from next month is to invite a model developer to give a seminar to ECHA. And if everything goes fine and we get the legal clearance, etc., we are also going to make this seminar for ECHA publicly available. There are a number of challenges in doing that. And one thing is that if the model is commercial, then it looks like we are endorsing or giving a commercial advantage to a commercial developer. So in this case, we are going to use the seminar is about a free tool. And it's also, we don't want to create the expectation that if something is presented in an ECHA seminar, it's automatically compliant and will be accepted, etc. So we will have disclaimers about it. But if everything goes fine, at least for some public tools, we may be able to invite the developers to give such information. Also keep in mind that many developers, they anyway publish articles, they present in conferences, etc. So maybe if it doesn't come from ECHA, you can find this information somewhere else.
And maybe to add to it, so we acknowledge that this clear request or clear need, because that was not the first question about it. We'll try to see what to do. But as Andrea mentioned, there were already quite few webinars before about the QSARs and QSAR toolbox as well. When you will find some as well hands-on example and as well concrete practical example how you can assess the reliability of predictions from most common publicly available models. So some of those information is already exists, and we will start to see how we can do it with the newer or with the other models in the future, and how we can as well demonstrate the QAF alliance. So we don't promise that we'll certainly do it and we'll cover everything, but we promise that we'll try to think how to address this need.
Thank you. I think I go to the biodegradation question. We had already some discussion on the global and local models, so I think that is at least partly covered. So since biodegradation is a very complex environmental procedure, how justified is the use of QSAR models for prediction of biodegradation rate of substances or mixture?
I think it's quite similar, the one that we had already addressed, that Andrea clarified. There is better models for ready biodegradability, but not for simulation studies. They can be used as innovative evidence, as supporting evidence, especially if you have multi-constituent substances or UVCBs. So then they can be really helpful to set up a testing strategy or to know where to focus your attention.
Okay, thank you. I think we have time for a few more questions. Maybe I take the question on how do you weight QSAR predictions and the QSAR result in dossier evaluation when you have multiple tests, outcomes, and results? Do you use only the QSAR results?
I think, Ulla, we answered this before, and we weren't sure about the interpretation of the question. I don't see any reply to that, so maybe the interpretation we gave before was good enough.
Okay. Then let's go. Okay, let's go to the question related to when or is it ever acceptable to make a qualitative assessment when the result of a highest or lowest extreme?
Mm-hmm. I think we have seen also models in the past. It depends again on the purpose. So I can imagine if you have if you have a question on classification, and this would be good enough, I could imagine that is a way to go. However, we often have a dual use, so it's the predictions are used for risk assessment or the safety assessment, and also for classification and labeling. And for the safety assessment, usually we are after a quantitative output. So you really have to check if this is if it's possible. And there is also one assessment element if it fits the endpoint that is required. So there you have to check if it fits.
Thank you, Doris. Maybe I take next the question on the assessment framework and the R6, the ECHA guidance. Will you update ECHA guidance on QSARs, so the ECHA guidance R6, to reflect the publication of the assessment framework?
Mm-hmm.
Who would like to?
I think we already covered this question to some extent with Andrea. So basically, we are not planning updating this guidance at the moment. We may only eventually, if we'll start to use the assessment framework in our decisions, we may just add the link to the QSAR assessment framework at the beginning of the guidance in the QSAR part. But we don't foresee need for substantial updates.
Okay. Thanks, Thomas. I think we have covered many of the questions, but I'm trying to find something that we have not discussed. Maybe will 3D similarity be acceptable or just 2D similarity based on models?
I think if I can take it, 3D models are in theory supposed to be even better than 2D models. We don't see that many under reach, but if you have 3D models and they are available and provide good results, or in this case, good similarity matrices, they are welcome to be used.
Thank you, Andrea. And then maybe here one more. What kind of weight will ECHA assign to the assessment elements for predictions? Will it be those default ones indicated in the checklists? Is it the same weight assigned to assessment elements for all endpoints, or will there be differences?
Okay, so I can take it. All assessment elements in the QSAR assessment framework, they are something that we check already now. So we are going to check all of them, which means that all assessment element is of high weight, and we do not have a priori distinctions depending on the endpoints.
Maybe I could add, because we are going to check all assessment elements, it means also basically any of these assessment elements could lead to an incompliance. However, the framework is quite flexible on how to assess each assessment element. So there we have some flexibility. So we do not expect now that everything is perfect. I mean, we are realistic. So I think we will not reject. We aim not to reject any prediction that is good enough. So just to keep that in mind.
Okay, thank you. I think we will have time one more question. So let's take the last one. Do the output files from the QSAR model need to be submitted, and are these sufficient to fulfill requirements to submit a QPRF?
Thanks for picking that. Can I answer this one? Because we have seen that many times, and this is really difficult for us because certain elements are just not in the output file. There is no reflection if the prediction is fit for purpose. Often it's missing if there are similar substances that are predicted well. So really the output file is normally not enough. So you always have to look at it and compare with the QPRF. And now we have also the 4 new principles for a QSAR prediction. Please check the assessment elements, what we are going to check, and add manually what is missing. And then I think it's for both parties, it will be much better, less work than overall.
Okay, I think we are running low on the time. We are closing the end of this session. So thank you to everyone who included questions and all of those who listened in today. In particular, thank you to our panelists, Andrea, Doris, and Thomas. As I mentioned, all questions received today and within the scope of the webinar will be answered in our questions and answers document that will be published soon on ECHA website. So thank you again for joining us and have a great rest of the day.